Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The 2017 Nobel Prize in Physiology or Medicine recognized Jeffrey Connor Hall, Michael Rosbash, and Michael Warren Young for their fundamental discoveries concerning the molecular mechanisms governing circadian rhythms. This essay undertakes a rigorous evaluation of the claims, arguments, and conclusions presented within their body of work, focusing on a section-by-section analysis of publications directly linked to their Nobel Prize. The aim is to dissect the core scientific advancements, assess the evidence supporting those advancements, and understand the enduring impact of their contributions on our comprehension of biological timekeeping. Section 1. Initial forays into the period gene and its protein product. Hall, Rosbash, and Young's early investigation centered around the period gene in Drosophila melanogaster. Their claims focused on demonstrating that mutations in period affected the fly's circadian rhythm, specifically its locomotor activity and eclosion, which is the emergence of the adult fly from its pupal case. These experiments, detailed in publications such as those appearing in Cell, involved meticulous genetic analyses, mapping of the period locus, and complementation testing of various mutant alleles. Evaluation of these claims hinges on the careful design of the experiments. For instance, the researchers presented compelling evidence that different period alleles resulted in altered rhythm lengths. One allele caused a shortened rhythm, another a lengthened rhythm, and a third resulted in a rhythmicity. They demonstrated the period gene resided on the X chromosome. Utilizing classical genetic mapping techniques, the crucial element of the initial claims was the assertion that the period gene itself was a central component of the circadian clock, a claim supported not through direct observation of its function, but through phenotypic analysis of its mutations. The evidence, based on these phenotypic observations, was robust. Each mutated period allele demonstrably caused a distinct alteration in the circadian rhythm, strongly indicating a direct link between the gene's function and the regulation of biological timing. Section 2. Cloning and characterization of the period gene. The subsequent step involved the monumental task of cloning and characterizing the period gene. This achievement allowed for a more in-depth investigation into its structure and expression. Publications during this period, also largely featured in Cell, report the successful isolation of the period gene using positional cloning techniques. Evaluation of these claims requires understanding the complexities of these techniques, which involved identifying overlapping DNA fragments contigs spanning the chromosomal region where period was located. The researchers utilized chromosome walks, a technique involving the iterative isolation and characterization of overlapping DNA fragments, thereby walking along the chromosome until the period gene was identified. Critical here is that the identification of the period gene was confirmed by showing that DNA fragments from the contig could rescue the aphysmic phenotype of period mutants when introduced into their genome. They also demonstrated that the period gene encoded for a novel protein of approximately 1,200 amino acids, which they termed PAR. The deduced amino acid sequence revealed no obvious homology to previously characterized proteins, suggesting that PAR had a unique function. They showed that the period messenger RNA level fluctuated rhythmically, reaching a peak at the end of the night and declining during the day. This finding provided the first indication that the period gene's expression was itself regulated in a circadian manner, adding another layer of complexity to the understanding of the circadian clock. The claim that the period mRNA levels cycled rhythmically was substantiated by northern blot analysis, a technique used to measure the abundance of specific RNA molecules. Section 3. The identification of TRM and its interaction with PAR. A significant advancement emerged with the discovery and characterization of the timer's gene TIM. Hall, Rosbash, and Young, in separate but complementary studies, demonstrated that TIM was essential for the proper functioning of the circadian clock. Specific publications being predominantly in cell, described the isolation of the timeless gene and the identification of its protein product, TIM. They then demonstrated a crucial interaction between PER and TIM, showing that TIM was required for PER protein to accumulate in the nucleus. Evaluation of these claims requires careful examination of the experimental evidence supporting the PER-TIM interaction. The experiments demonstrated that in timeless mutants, PER protein levels were dramatically reduced, suggesting that TIM protected PER from degradation. Koimanot precipitation experiments provided direct evidence that PER and TIM physically interact, forming a complex. This complex formation was shown to be essential for the translocation of PER into the nucleus, the site where it was thought to exert its regulatory function. This discovery was central because it indicated that PER did not act alone but required TIM as a partner to fulfill its role in the circadian clock. The identification of TIM and the revelation of its interaction with PER constituted a major breakthrough, moving the field closer to understanding the molecular mechanisms governing circadian rhythms. Section 4. The model of the negative feedback loop. The culmination of these findings led to the formulation of a model describing the molecular basis of the circadian clock, specifically a negative feedback loop involving PER and TIM. This model, presented in various publications culminating in comprehensive reviews, proposed that PER and TIM formed a complex that inhibits the expression of their own genes, period and timeless. Evaluation of this claim necessitates understanding the evidence supporting this model. The experiments demonstrated that PER and TIM proteins accumulate in the cytoplasm during the night, translocate to the nucleus, and then decrease the transcription of period and timeless genes. This repression of transcription was hypothesized to occur through direct or indirect interactions with transcription factors. As PER and TIM levels decline, the repression is relieved, allowing period and timeless genes to be transcribed again. This cyclical pattern of gene expression and protein accumulation constitutes the core of the negative feedback loop. One crucial piece of evidence supporting this model was the demonstration that PER protein combined to the period promoter region, suggesting a direct role in repressing transcription. However, they also acknowledged that other factors might be involved in the repression, as PER binding to the promoter was not always sufficient to fully inhibit transcription. The model, while initially focused on Drosophila, provided a framework for understanding circadian clocks in other organisms, including mammals. Section 5. The role of double-time kinase. The complexities of the circadian clock mechanism were further illuminated by the discovery of double-time DBT, a sewing through amine kinase that phosphorylates PER. This discovery, documented in numerous publications, revealed that DBT regulates the stability and turnover of PER. In particular, DBT phosphorylation targets it for degradation. The claims regarding DBT's role in regulating PER stability are evaluated by scrutinizing the experimental evidence demonstrating the consequences of DBT mutations. Mutant DBT isoforms exhibited altered kinase activity and caused changes in the cycling of PER. Experiments showed that specific mutations in DBT resulted in increased PER levels and slowed down the circadian clock. These findings indicated that DBT was crucial for the timely degradation of PER, contributing to the appropriate cycling of PER protein levels. This discovery added another layer of complexity to the feedback loop, highlighting the importance of post-translational modifications in regulating circadian rhythms. It also provided a mechanism for explaining differences in rhythm length observed in different period alleles. Variations in per amino acid sequences affected DBT phosphorylation, leading to variations in PER stability and, consequently, rhythm length. This discovery underscored the intricate nature of the circadian clock, highlighting the interplay between gene expression, protein interactions, and post-translational modifications. Section 6. Cryptochrome and light input. The investigation of light input into the circadian clock revealed the role of cryptochrome CRY, a blue light photoreceptor. Publications focused on cryptochrome's function in synchronizing the clock to the external environment. These reports indicate that CRY absorbs blue light and triggers a signaling pathway that leads to the degradation of TIM, thereby resetting the clock. Evaluation of the claims regarding CRY's role in light input necessitates a careful assessment of the experimental evidence. Flies lacking cryptochrome were shown to be insensitive to light and exhibited impaired circadian responses to light dark cycles. Furthermore, experiments demonstrate that exposing flies to blue light resulted in the degradation of TIM in a CR-independent manner. This degradation of TIM resets the clock by allowing the period gene to begin transcribing, restarting the entire feedback loop. The discovery of CRY provided a critical link between the external environment and the internal circadian clock. Light, the primary synchronizer of circadian rhythms, acts through CRY to adjust the phase of the clock. This allowed for a more complete understanding of how organisms adapt to daily environmental changes. The insight was supported by the demonstration that CRY interacts with other signaling molecules, forming a complex that mediates the light-dependent degradation of TIM. Section 7. The mammalian counterparts and homologies. The discoveries in Drosophila paved the way for understanding circadian clocks in mammals. Hall, Rosbash, and Young's pioneering work led to the identification of mammalian homologs of period, timeless, and other clock genes. These findings, published in high-impact journals, demonstrated that the basic principles of the circadian clock are conserved across different species. Evaluation of the claims concerning mammalian clock genes requires an assessment of the evidence demonstrating the similarities and differences between the Drosophila and mammalian systems. The identification of mammalian period genes PER1, PER2, and PER3 was based on sequence homology to the Drosophila period gene. These mammalian PER proteins were shown to exhibit similar rhythmic expression patterns and to interact with other clock proteins, further supporting their role in the circadian clock. Although a direct homologue of Drosophila thymus was not found in mammals, other proteins, such as AY1 and AY2, were shown to play similar roles in the mammalian clock. These CRY proteins were found to bind to PER proteins and to inhibit their activity, contributing to the negative feedback loop. While the mammalian clock operates on similar principles as the Drosophila clock, there are also significant differences. For example, the mammalian clock involves a more complex interplay of multiple PER and CRY proteins, as well as other clock components. Nevertheless, the fundamental concept of a negative feedback loop remains conserved, underscoring the evolutionary importance of this mechanism in regulating circadian rhythms. Section 8. Transcriptional regulation, clock and cycle BMAL1. Further research broadened the understanding of the mammalian clock by identifying the clock and cycle proteins, later renamed BMAL1, which are basic helix-loop-helix transcription factors. Applications focused on clock and BMAL1 demonstrated that these proteins form a heterodimer that binds to EBOX enhancer sequences in the promoters of period and other clock genes, activating their transcription. Claims regarding clock and BMAL1 acting as transcriptional activators need careful evaluation of the in vivo and in vitro data. Experiments showed that clock and BMAL1 proteins accumulate in the nucleus and bind to EBOX elements in the promoters and genes, activating their transcription. Mutations in clock or BMAL1 resulted in disrupted circadian rhythms, confirming their crucial role in the clock mechanism. The discovery of clock and BMAL1 provided a clear mechanism for the rhythmic transcription of period and other clock genes. The clock BMAL1 heterodimer acts as a positive regulator, driving the expression of genes that are subsequently repressed by PER and CRY proteins. This positive and negative feedback loop is essential for generating sustained circadian oscillations. The identification of clock and BMAL1 and their role in transcriptional activation significantly advanced the understanding of the molecular mechanisms underlying the circadian clock in mammals. Section 9. Post-translational regulation in mammals. In mammals, post-translational modifications, particularly phosphorylation, play a crucial role in regulating the stability, localization, and activity of clock proteins. The demonstration that kinases, such as casein kinase 1 epsilon, CK1 epsilon and others, phosphorylate proteins and regulate their degradation, rate significantly contributed to understanding the clock. Examination of publications focusing on kinases like CK1 epsilon requires a thorough assessment of the data linking phosphorylation to protein stability and function. Experiments demonstrated that mutations in CK1 epsilon, or mutations in perphosphorylation sites, altered per stability and circadian rhythm. CK1 epsilon phosphorylation of PER was shown to target PER for degradation via the ubiquitin proteasome pathway. The post-translational modifications of PER and other clock proteins are highly regulated and influence the period and robustness of the circadian clock. The discovery of post-translational regulation added another layer of complexity to the clock mechanism. Many different kinases and phosphatases are involved in regulating the phosphorylation state of clock proteins, providing numerous opportunities for fine-tuning the clock's period and amplitude. Section 10 Feedback loop interlocking and network complexity. It became clear that the circadian clock is not a simple feedback loop, but a complex network of interconnected loops. The identification of additional feedback loops involving other clock genes and proteins expanded the understanding of the clock's robustness and adaptability. Assessing publications that detailed these additional feedback loops requires scrutinizing evidence demonstrating the interconnectedness of various clock components. These reports highlight the existence of secondary feedback loops that regulate the expression of clock-controlled genes, which in turn affect the core clock mechanism. For example, the REVERB-alpha and ROR-alpha transcription factors, which are themselves regulated by the clock BMAL1 heterodimer, bind to other elements in the genome and regulate the expression of other clock genes. The interlocking feedback loops provide redundancy and stability to the circadian clock and allow it to adapt to different environmental conditions. The complexity of the clock network also allows the clock to integrate different signals from the environment, such as light, temperature, and food, to coordinate various physiological processes. Section 11 Clock controlled genes and physiological outputs. The circadian clock regulates a vast array of physiological processes, from sleep-wake cycles to hormone secretion and metabolism. Identification of clock-controlled gene CCGs and their tissue-specific expression patterns expanded the understanding of the physiological outputs of the circadian clock. Validation of the claims connecting clock-controlled genes with specific physiological outputs requires evaluating the evidence that links clock gene expression to downstream processes. Experiments using gene knockout mice and other genetic manipulations demonstrated that disrupting the circadian clock leads to profound changes in physiology. For example, disruption of the clock gene BMAL1 results in metabolic dysfunction, immune system abnormalities, and increased susceptibility to disease. The clock controls the expression of a large number of genes in a tissue-specific manner, allowing it to coordinate different physiological processes in different parts of the body. This is crucial for maintaining overall health and well-being. Understanding the physiological outputs of the circadian clock has important implications for human health and disease. Disruptions in circadian rhythms have been linked to increased risk of sleep disorders, metabolic disorders, cardiovascular disease. Section 12 Clinical implications and therapeutic applications. The knowledge gained from studying circadian rhythms has led to new insights into the pathogenesis of various diseases and to the development of new therapeutic strategies. The exploration of the clinical implications of circadian research and the potential applications of circadian-based therapies necessitates critical examination of the clinical evidence. Evidence from clinical trials and epidemiological studies demonstrated that manipulating the circadian clock can improve the outcome of various diseases. For example, chronotherapy, which involves administering drugs at specific times of day to maximize their efficacy and minimize their side effects, has been shown to be effective in treating cancer and cardiovascular disease. Light therapy and melatonin supplementation are used to treat sleep disorders and other conditions associated with circadian rhythm disruption. The development of new drugs that target clock proteins is also a promising avenue for future therapeutic interventions. Circadian medicine is a rapidly growing field that holds great promise for improving human health. Section 13, Evolution and Conservation of the Clock Mechanism. The conservation of the circadian clock mechanism across diverse species suggests that it evolved earlier in the history of life. The claim that the core features of the clock are conserved across different organisms must be supported by comparative genomic and biochemical evidence. Comparing the clock genes and proteins from different species reveals a high degree of similarity, indicating that the basic principles of the circadian clock have been maintained over evolutionary time. The circadian clock is found in bacteria, fungi, plants, and animals, suggesting that it evolved very early in the history of life. The evolutionary conservation of the circadian clock underscores its fundamental importance for survival and adaptation. Organisms with a functional circadian clock are better able to anticipate and prepare for daily environmental changes, such as changes in light, temperature, and food availability. This allows them to optimize their behavior and physiology for survival and reproduction. Section 14, Future Directions and Unanswered Questions. Despite the tremendous progress that has been made in understanding circadian rhythms, many questions remain unanswered. The assertion that the path to full understanding is still ongoing and there remain avenues to explore is supported by current literature. Major areas of ongoing research include identifying new clock genes and proteins, understanding how the clock interacts with other cellular processes, and developing new therapeutic strategies for treating circadian-related disorders. One major challenge is to understand how the circadian clock integrates different signals from the environment. Another challenge is to understand how the clock controls the expression of such a diverse array of genes in a tissue-specific manner. The precise mechanisms by which the clock regulates metabolism, immunity, and other physiological processes remain largely unknown. Future research will likely focus on addressing these unanswered questions and on translating the knowledge gained from basic research into new therapeutic applications. In conclusion, the work of Hall, Rosbash, and Young has fundamentally transformed our understanding of the molecular mechanisms governing circadian rhythms. Their meticulous genetic analyses, biochemical characterizations, and elegant experimental designs have revealed the core components of the circadian clock and how it functions to regulate physiology. By elucidating the negative feedback loop involving PA, TIM, and other clock proteins, they provided a framework for understanding circadian clocks in organisms ranging from flies to humans. While their research primarily focused on Drosophila, their findings have had a profound impact on the field of chronobiology and have led to new insights into human health and disease. Their discoveries are of lasting importance, not only in the field of biological timing, but also in the broader context of molecular biology and genetics. The recognition they received through the Nobel Prize was a fitting acknowledgement of their remarkable contributions to science. They have illuminated a fundamental process that governs life on Earth, demonstrating how organisms adapt to the daily rhythms of our planet. The work also demonstrates, with a touch of irony, that a deep understanding of even a simple fly can unlock secrets applicable to complex human biology.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.